Welcome back to Particle Pharma. In the previous session, I have discussed about meiosis 1 and today I will be discussing about meiosis 2, which is also again divided into four phases: prophase 2, metaphase 2, anaphase 2 and telophase 2. Meiosis 2 is similar to mitosis where recombination is not taking place. Or in short, the cells are dividing by itself. Recombination is not taking place in meiosis, where it leads to a formation of four daughter cells which are genetically diverse, means they are different from each other. Why? Because due to individual assortment and genetically diverse recombination which has taken place in prophase 1 of meiosis 1, and they are formed from diploid parent cell. So in prophase 2 of meiosis, we can see there will be a formation of spindle fibers from centrosome, and there will be a nuclear membrane breakdown so that the chromosomes can be easily available to the spindle fibers. Let me move to the second phase of prophase meiosis 2 is metaphase 2. In metaphase 2, we can see there will be a chromosome alignment again at the center line, which is an imaginary line, that is metaphase 2 plate. There is no such thing as line in the cell, just it is an imaginary line. Along with that, the sister chromatids or condensed chromosomes align at the center. And spindle fibers, what they will do is that have arisen from the centrosome, they will get attached to the sister chromatids so that they can pull them back to the poles. And this is a metaphase plate. So it will lead to a next phase, that is telophase. Sorry, anaphase 2. In anaphase 2, what will happen is sister chromatids are pulled apart, which are there in the center or at the metaphase plate. Now they have moved towards to the pole of the cell, and this separation is done by spindle fiber. They will get shortened and they will pull the chromosome to one end. And the haploid nature of the cells are maintained in this anaphase 2. And anaphase 2 will check that each daughter cell should get complete set of chromosomes, neither extra nor less. And they will also check that their cells should be free from duplication or deficiency, and there should be a proper division of genetic material. So the genetic material which are present in the chromosome form, they should get properly divided. That is checked in anaphase 2. So there will be a beginning of formation of four daughter cells. Just it has begun, but they are not completely divided. They will get divided in telophase 2. Not completely divided, but they will begin to divide. In telophase 2, there will be a formation of nuclear membrane and spindle fibers will get dissolved and the nuclear membrane form, and they will cover the chromosomes so that each individual should get the correct number of chromosomes, and there will be a formation of furrow so that cells should get divided, and from that point only cytoplasm will start to divide, and it leads to a formation of four daughter cells, and it should contain a single set of chromosome. Single set of chromosome can be visible in each cell. So it leads to a formation of daughter cell, and there will be it is the last stage of the meiosis 2. So in cytokinesis, what will happen is, as we have known that the two daughter cells are formed from previous single daughter cell. So here two daughter cells are formed, here two daughter cells are formed. Total four daughter cells are formed which are genetically diverse, which I have already discussed. And nucleolus and nuclear membrane, again they will reappear. Sorry, it's a mistake here. Nuclear membrane is this one. And nucleolus, so they will start to reappear. So by this, meiosis 2 is completed, and from now on, I'll be starting the other topics, which I'll be telling you later on which topics I'll be starting. If you people have any wish to tell that which topic should I start, please just let me know. So after cytokinesis, I just want to give a brief, what I can say, brief review on significance of meiosis. So I'll just complete within minutes. Why meiosis is required according to us? As it produces a haploid gametes. So from diploid to haploid nature, so that they should not be polyploidy that is observed in offspring. What will happen is, if organism is going to get divided through mitosis, just imagine there was no meiosis, organisms are going to get divided by mitosis. What will happen? There will be an increased number of chromosomes per every offspring. Again it will lead to polyploidy, again it will lead to formation of superspecies. So that's why meiosis is required. Next is genetic variation, that is independent assortment and crossing over that has taken place in prophase 1 of meiosis 1, where sister chromatids, they will exchange the genetic material through chiasmata and they will form bivalent. And independent assortment, which means there will be a randomly arrangement of chromosomes at the metaphase plate. So by this, there will be a formation of new traits, or that will leads to a formation of new traits or new offspring. Not new offspring, I mean to say that new habits in the offsprings, which leads to adaptation to evolution. Next is prevent disorder. So as increase in chromosome number is not taking place due to meiosis, it will prevent polyploidy, which I have already told you, and it also helps in maintaining same number of chromosome in all the cells. And from diploid to haploid nature is forming, and this haploid cell from another offspring, they will get fused and they will form a new offspring. And if polyploidy is seen in this offspring, again it will leads to disabilities or disorders, that is prevented by meiosis. And we all know that it is helping in the formation of gametes, that is male and female gametes, which are required for sexual reproduction. Next is ability to adapt to environmental change or enhance species survival, which I have already discussed here. Due to that, what will happen, that they will allow to produce, or meiosis will allow to produce traits with super, I can say, abilities, or abilities that are helping them to adapt to environment change that we can see in day-to-day life, so that they can cope up with environment. So by this, I have completed the meiosis. So in my next sessions, I'll be starting the other topics that you will come to know soon. Till then, stay tuned and subscribe to our channel, and you can check the videos related to cell and cell division on our Instagram page, that is in our highlight session. Okay, and thank you so much. If you people found any mistakes, please feel free to contact us. Till then, thank you so much.